Okay, so let's come back to the slides. I think this slide we already finished, right? Last time, HDL cycles, or the reverse cholesterol transport process, and this 1, 2, 3, 4, 5 proteins are very important. Try to remember, and these 5 proteins that involved in the HDL cycle, right? And HDL cycle means that 2 different versions of HDL, alpha HDL and beta HDL, they are converted, but depend on the lipid core, right? And these 2 type of HDL have a different function. So beta HDL basically is empty, right? The empty sort of like a vehicle or a trunk, and mostly the abundant in the the peripheral tissue, right? So the purpose of the beta HDL basically pick up the the free cholesterol and phospholipids from the peripheral tissue cells and transported the free cholesterol and cholesterol from the peripheral tissue back to the liver. And once the beta HDL picks up the free cholesterol, phospholipids from peripheral tissue and then enter the circulation or plasma, and then through the several steps, so the beta HDL eventually can convert it to alpha HDL. So alpha HDL eventually takes the free cholesterol from peripheral cells to the liver for the disposal, right? And this 5 proteins that are important that regulates this whole process HDL cycle. And this gives you a little bit detailed the whole process. So as you can see here, the peripheral tissue cells, particularly like artery wall, like macrophages, then and the other side represents the liver. So in the between is the things that happening in the plasma, right? So ABCA1, which is a transporter, right? Remember, it's located on the cell surface of a peripheral cells, and the main function of ABCA1 is to transport free cholesterol from peripheral cells and upload the free cholesterol to the beta HDL, right? And once they're receiving the the free cholesterol from peripheral tissues, the beta HDL eventually converted to so-called discoidal HDL. So discoidal HDL eventually enter the circulation plasma, and during in the plasma, there are several steps, right, occurs. The first of all, the reacidification of free cholesterol, right, in the discoidal HDL, and this is enzyme LCAT. It's an enzyme that catalyzes reacidification of free cholesterol within the discoidal HDL. And once the free cholesterol is acidified to cholesterol ester, so cholesterol ester then can enter and then can internalize to the core, to the lipid core, right, of the HDL. So HDL eventually becomes the discoidal HDL becomes the alpha HDL. And also the so-called lipids exchange, right, occurs again in and also in the plasma between alpha HDL and VLDL, IDL, and LDL. So this lipid exchange usually is catalyzed by the protein called CETP, right? So CETP is a protein. It's also present in the plasma, and this protein, what this protein does is to facilitate the lipid exchange between the different lipoproteins. Particularly, the cholesterol esters is transferred from alpha HDL to VLDL, IDL, or LDL. So makes the LDL more rich in the cholesterol esters. And in the meantime, the triglyceride is transferred from these three lipoproteins into alpha HDL, right? So eventually the lipid core in the alpha HDL contains majority actually of the cholesterol ester, but also some triglyceride, right? So eventually the alpha HDL being picked up by the liver. So for further metabolize, so cholesterol can be used to synthesize bile acid and also steroid hormone in the liver. So this is the sort of the cholesterol reverse cholesterol transport process. And once the lipids is picked up by the liver, the alpha HDL then become beta HDL, right? Because you almost depleted all the lipids. And then the beta HDL then go back to the peripheral tissue and take the free cholesterol again from the peripheral tissues, right? So and these proteins are very important that involved in the the whole process of HDL cycle. And this lipid exchange is also very important for formation of LDL. Remember, one LDL initially is converted from VLDL, right? So it's more has more triglyceride, less cholesterol. So after exchange, the lipids exchange, then LDL become more cholesterol-rich lipoproteins. Any questions about this HDL cycle? Right? Okay, so now more information. So what happens to alpha HDL when it's become beta HDL? So first of all, the reduction of HDL core volume, right, because of the cholesterol esters being picked up by the liver, so the volume, lipid core volume, is reduced when alpha HDL is converted to beta HDL. And secondly, the dissociation of ApoA1 with alpha HDL. So so ApoA1 is gone. And the third is internalization of cholesterol esters from alpha HDL. And finally, the hydrolysis of triglyceride in the alpha HDL by the liver, like hepatic lipase. So all these things actually they're happening when the alpha HDL is converted to beta HDL. Right? Yes.
I'm confused. Why does it reduce? Why is the core volume reduced? Because isn't it going from like?
Yeah, because it's cholesterol, right? It's picked up by the liver, So then alpha HDL is losing the cholesterol esters, right? So the volumes decrease. Right, so there's something happening in the liver, right? The conversion of HDL to beta HDL. And the conversion from beta HDL to alpha HDL, this is a process that occurs in the peripheral tissue, right? Peripheral tissue have a very the beta HDL is very abundant in the peripheral tissue, so then picks up the free cholesterol. So what happens to the beta HDL when it's converted to alpha DHA? Basically, it's beta HDL is enriched, right, in the peripheral tissue, and picks free cholesterol or phospholipids from peripheral tissue. And again, the ABCA1, this is transporters. It's very important that control free cholesterol efflux to beta HDL in the peripheral cell. Efflux means that transport free cholesterol from the cell and upload it to the beta HDL, right? And eventually beta HDL will receive enough free cholesterol or phospholipids forming the discoidal HDL, and then re-enter the plasma, and for the further metabolism. All right, so the other thing is happening in the plasma, that the alpha HDL. Once alpha HDL, get the enough free cholesterol and phospholipids from the peripheral cells, and in the plasma, the free cholesterol need to be re-acidified to cholesterol ester in order to be internalized to the core, to the inside of the HDL, right? So the enzyme is called LCAT, right? It's catalyzed this conversion, and then the also exchange of lipids like triglyceride and cholesterol between alpha HDL and the other type of lipoproteins also important, to allow the alpha HDL to get more cholesterol esters from the other lipoproteins, and in the meantime to give the triglyceride to the other lipoproteins. So eventually the cholesterol is loaded to the HDL, and alpha HDL will go to the liver, and liver picks up alpha HDL and hydrolyze the cholesterol ester inside of alpha HDL, right? So inside of the liver, the cholesterol esters can be further metabolized and utilized for synthesized bile acid and excreted, right? And or can be utilized for synthesis of steroid hormones. So this again is a very important way to get rid of the extra cholesterol in the peripheral tissue, right? Actually, I have another video, but I'm not going to play it. You guys can just watch noodles, right? All right. So something that you need to know about the lipoprotein. First of all, the major component of function of the four lipoproteins, like chylomicron, VLDL, LDL, and HDL, right? So try to remember what is the core, lipid composition in the core, right, of these four different lipoproteins, and what are the apoproteins on the surface of these four different lipoproteins, and how they are synthesized, or where they are they synthesized is very important, and what is the function of each lipoproteins. And the other thing is important is the role of LDL receptors, right? LDL receptor, that is very important, regulates the LDL uptake, and eventually can control the LDL cholesterol levels in the circulation. And factors that I just mentioned, or enzymes that involve in the metabolism lipoproteins, like particularly the HDL cycles, like ACAT, LCAT, ECPT, ABCA1, and SRB receptors, how they regulates the cholesterol transport, reverse transport, how and eventually how they may affect the cholesterol levels and the risk for the atherosclerosis, right? And the last thing that you guys probably also wanted to make sure that be able to distinguish the functions of LPL, hepatic lipase, HSL, right? So I can't remember if I mentioned this, that if you compare the difference between LPL and HSL, did I mention? Probably not, right? So LPL basically is secreted or secreted from the adipose tissue or muscle, right? And the main function of LPL is hydrolyze the triglyceride in both chylomicron and the VLDL, right? And HSL is the key enzyme. Guys still remember? In which pathway? Metabolic pathway? The key enzyme for which metabolic pathway? Lipolysis, right? Okay, so HSL is also hydrolyzed triglyceride, but it's inside of the fat cells, right? Inside of cells. So this is the one thing that is different. The LPL hydrolyzed triglyceride in the circulation in the chylomicron and the VLDL. The HSL hydrolyzed triglyceride inside of cells. And second thing is difference is that the regulation is also different. If we're thinking about just in the adipose tissue, so LPL basically is increased during the fat condition, right, and can be stimulated by insulin, and HSL is regulated by catecholamine, epinephrine, during the fasting condition, right? And hepatic lipase is the lipase in the liver, so hydrolyzed triglyceride in the liver. Okay. Any questions? Sorry. Yeah, we do have. So I think we finished this. Okay, so the last section of lipid metabolism, regulation lipoprotein metabolism and clinical relevance. And this just gives you a better idea, I think, to see different lipoprotein, the composition of the lipoproteins. Again, the triglyceride and VLDL are the triglyceride-rich lipoprotein, right? And these two lipoproteins basically is regulate the triglyceride metabolism, right? So transport the triglyceride from intestine to the liver. That is the function of the chylomicron, and VLDL is transport triglyceride from liver to the other tissue like adipose tissue. And HDL, LDL are the cholesterol-rich lipoprotein and play an important role in the cholesterol levels and metabolism. And particularly HDL, right, is considered the good cholesterol, and it's very important to get rid of the extra cholesterol in the body to prevent the deposition of cholesterol, right? And this slide just put all the lipoprotein together. I hope not make things more complicated, but I think it's probably important, to have a comprehensive understanding all these four different type of lipoprotein, right? They actually work together. They actually also have, a relationship in terms of the regulation of lipoprotein metabolism. So, for example, in the fat condition, right, we absorb lots of lipids, and these lipids need to be transported into the body, into the tissue, particularly in the liver, the adipose tissue and liver. So chylomicron is the lipoprotein that's synthesized in the small intestine, and then carry the triglyceride primarily, and also some cholesterol esters from small intestine and to the adipose tissue for storage, right? And LPL, again, is very important. Lipase that's synthesized and secreted by adipose tissue, and hydrolyzed triglyceride in the chylomicron, and release fatty acids, glycerol. The fatty acids can be taken up by adipose tissue, adipose cell, and inside adipose cells, and free fatty acids be reacidified to triglycerol and stored in the adipose tissue as the energy. So during the fasting, and in the meantime, because of the majority of the triglyceride has been taken up by the adipose tissue, so chylomicron become chylomicron remnant, and chylomicron remnant eventually is taken up by the liver, right? And also, we absorb the lots of the glucose, carbohydrate, right? And we always know. This always happening during the fat condition, particularly when we consume a high carbohydrate diet. So, glucose can be converted to fatty acids through the lipogenesis pathway, right? And fatty acids reacidify to triglyceride. And this triglyceride also need to be made in the liver, but also need to be transported to the adipose tissue. So the triglyceride being uploaded to the VLDL. So VLDL basically is made in the liver, right? And similarly to chylomicron, LPL also is needed to hydrolyze triglyceride in VLDL, right? And then for release fatty acid for pickup, by adipose tissue. There's something happening in the fat condition, and once the lots of triglyceride being picked up by the fat tissue, the VLDL has become IDL and eventually LDL, right? So you can see the LDL if you compare the core lipids between LDL and VLDL. So LDL has much less triglyceride and more cholesterol. And this cholesterol ester was actually initially comes from alpha HDL, right? So after lipid exchange, so lots of cholesterol esters being transferred to the IDL and VLDL from the HDL, and the triglyceride being also further picked up by the HDL. So eventually LDL will be picked up by the peripheral tissue or the liver, both liver and the peripheral tissue. And LDL receptor is required, right, for pickup LDL. And HDL, the beta HDL is converted from alpha HDL when all the core lipids is picked up by the liver, right? So beta HDL then goes to the peripheral tissue and picks up free cholesterol, phospholipids, and convert it back to the alpha HDL. So alpha HDL is lipid-rich HDL, right? And then takes lipids from peripheral tissue, particularly cholesterol esters from the peripheral tissue, and go back to the liver, and deliver all the cholesterol esters from the peripheral tissue to the liver for disposal. So this is a sort of all things happening during the fat condition, and so the 4 different type of lipoprotein all involve all this, the metabolism, the cholesterol, triglyceride metabolism, right? And this is, so now let's talk about regulation of lipoprotein metabolism. We know regulatory factor always involve genetic, dietary, and environmental, right? It's the main 3 main factors that involve, the lipid metabolism. So I guess should we need a break? Okay.
She got scared that they never did.
They cut me everywhere.
Nice.
Do you remember that? We used to play that all the time. Yeah. Do you remember that? Crisscross, spiders crawling up your back, spiders crawling down your back. Big spoon. Now you've got the shiveries. I have shiveries. Oh, you. I'm gonna get some shit for the party no. Everything is thunderstorm because I'm somehow I was so wet and then I was so sick of being wet.
When we had concerts, like I had a bunch of
but there's only so much you can take though before you're cold and wet still. It's coming down sideways. What is going on?
It's the one who lived downstairs or like on the main level. Like
Everyone is mad at everyone right now. I don't know what to do. Oh my god, this is so hilarious. And me out here. So anyway, so me out here. So I take on everything. I'll take her green and figure out whatever. So I said,
Why is she take
no, this is so hilarious. So I texted and I doesn't hit the notice. So I hope this is maybe all the stuff are going on. While you're making a fun to listen. Oh yeah. I know, I didn't like it. Would you be cool if being a turn?
Wait, is this what you're gonna say in the group?
Yeah, I would let you know because all of them didn't like but one. And it's like you gotta agree, you'd be cool if being a turn. If you don't want to go to school, like I'm sure you're because like people are gonna come out of each other and be like, okay, you're thirsty, like I'm like, and I'm like, yeah, that would be
No, you just said some booze maybe for yes.
That's something I'm going to be no one
No.
and then it's like I don't become good, but if you don't have any, I answered that time, I'll send it each, and then she's like, oh, I'll send this and then or if you want to make fun, because she's like, they need to they don't get it. So I was like, I'll tell you when I contribute to that. And this is so hilarious. So I said that, we need to respect the show. And I was going to text back. I said, sounds good, I was going to all the time.
Nina's the middle one, the one who lives on the main.
Yeah, I'm thinking about it.
She's the one who lives on the main. Yeah, we already decided we want to do this.
And then of course, it's like literally, I guarantee you, I was setting this up way before holiday. And then I was like, I didn't put something that always takes so much math. That's kind of me, which like that's true. Like, I'm fine with that. I'm just like, I don't even know what to say right now because we had some complication holiday. And then my own parents or something like this. I get that. And she's getting paid too, right? Like, it's not just one person. It's like, I'm going to spend money on being
When me and Olivia hosted our like Christmas and Christmas, just with like 10 other people, and we tried all the food, it came up to like almost $20. So like, I know how that shit goes. And we didn't throw a party. We had a Friendsmas and a Friendsgiving, and we had like 10 people over. But like to provide for everybody, and then we have people like bring desserts or something. It still comes out to you have to cook a lot.
It's like I'm not like we're not planning on being like, oh, I want that music, drink everything.
No,
But if they have something, like I'll probably get maybe.
people are gonna come and go because there are gonna be other things. You know what I mean? Like they're not gonna just sit at your house and like it's homecoming. They have shit to do at home.
Right. So I'm like, I'm okay with my big head, and then I can go. I guess we're not doing that anymore.
That is throwing some major slide in that shit.
Yeah, but then you go, okay, I don't even really think of course, she always says be it. We're not all going to do it. Which that's true. Because I'm like, right, I'll probably spend maybe, who knows how much I'm going to spend on my shit like that. And then
Yeah, they'll just like stay on the side of the
I'm hitting up. I like spicy. I fucked up. Like I couldn't have leave when I was like I fucked up. Writing bigger, so I'm here with can't write in class right now. I'm just like, so your mom never would.
All right. Okay, so now let's talk about dietary regulation of plasma lipids. So two type of factors like dietary fat, the composition of fatty acids is more important, like whether it's saturated fatty acids or unsaturated fatty acids, right? So dietary fat basically regulates the cholesterol levels, both cholesterol levels and the triglyceride levels, right, in general. But dietary carbohydrates usually regulates the more, or primarily I would say regulates triglyceride levels, right? So, dietary composition is a little bit different in terms of their role or their effect on the lipids metabolism in the plasma. So again, dietary fat can regulate both cholesterol and the triglyceride levels, and dietary carbohydrate primarily regulates triglyceride level, not much cholesterol level, right? Which makes sense, right? Carbohydrate can be converted to fatty acids and triglyceride, right? If we, eat too much carbohydrates, mostly affect the triglyceride level. And so factors that can affect the plasma lipids, the more information, right? So the percentage of total calorie consumed as a fat, like fat portion, if I have a higher portion of fat in the diet, that is is a major contributing factor that regulates lipase level. And secondly, the percentage of a saturated versus unsaturated fat, right, is also important. Not just the total fat, but saturated, unsaturated fat is also matters. And then lastly, the amount of dietary cholesterol itself is also regulates, right, cholesterol levels. And we know the cholesterol levels is very important. The substrate, right, can inhibit the key enzymes. This guys still remember for cholesterol synthesis? HMG-CoA reductase, right? So cholesterol level, high cholesterol level basically inhibits the HMG-CoA reductase, so they inhibit cholesterol synthesis. And the major effects, both circulating cholesterol levels and circulating triglyceride level, can be all affected, right, by the what do we eat, right? What the total amount of fat and the composition of the fatty acids, and also amount of cholesterol in the diet. So all can regulate or affect the plasma lipase levels, basically cholesterol and triglyceride. You have a question?
Will you just repeat with the CEPT? So with the dietary cholesterol, it inhibits it the more you eat, or stimulates it?
Oh, you mean the dietary cholesterol? Well, actually, what I'm saying is the cholesterol level inside of cell, right? Inside of cell. So if cholesterol level is high inside of cell, that can inhibit the HMG-CoA reductase, right? So inhibits the cholesterol synthesis. But dietary, consume of the the cholesterol, but at some level will also affect the cellular levels of cholesterol, right? So this is something about the average of the desirable dietary sources of energy. So like the desirable that we would like to have, for example, carbohydrate, 55% of carbohydrate in our diet and 30% of fat in the diet. But in reality is that we usually have 42% of carbohydrate and a little bit high 40% of fat in our diet, right? And for the fat, we will wish we have like saturated fatty acids only account for 10%, but we actually have higher than 10%, like 17% basically in our diet. And also we have a 2 trans fat in our diet. We wish to have a 0 trans fat, or actually 1%, right? And then the carbohydrate, most of carbohydrate in our diet is actually the sucrose, right, which is not good. We hope we'll have a more percentage of a starch as a carbohydrate in the diet. All right. So now let's talk about the genetic factors that regulate plasma lipids, triglyceride levels, right? So some patients, they have ApoC2 deficient. What is the function of ApoC2? Do you guys remember? How ApoC2 deficient affect the cholesterol levels or lipoprotein levels. What is the function? What is the role of ApoC2? Which lipoprotein has the ApoC2 on its surface? Do you remember? Chylomicron, yeah, and VLDL, right? So what does ApoC2 do? Remember that very important enzyme, the activity requires ApoC2. We almost every session we talk about. Oh yes. Yeah, LPL, right. LPL, lipoprotein lipase, right? So ApoC2 is required for LPL activities, right? So what LPL does? Yeah. Right. So hydrolyzed triglyceride in where? In chylomicron and VLDL, right? Okay. So what happened to triglyceride levels? Increase, right? So if the patient do not have enough ApoC2 or ApoC2 deficient, the patient will have a higher triglyceride levels. You have a question?
In the blood?
Yeah, in the blood. Yes, in the circulation, right, in the plasma. And what about chylomicron and VLDL levels? If ApoC2 is not enough or it's not, it's also up, right? I mean, because you cannot efficiently hydrolyze triglyceride in VLDL chylomicron, so the triglyceride inside the chylomicron VLDL cannot be removed, right? So the chylomicron and VLDL just remain, in the circulation. So again, ApoC2 deficiency causes increased triglyceride levels, increased chylomicron and VLDL level in the plasma, in the circulation. Yeah.
And how is that diagnosed? Like, how do you diagnose ApoC2 deficiency?
That's a good question. How to diagnose the ApoC2 deficiency? Well, I'm not 100% sure, but I can guess. Maybe we can, for example, isolate the lipoproteins from the blood, right, and then measure the ApoC2 levels or something like this, so and see if ApoC2 level is normal or decreased or something like this. Okay, so now let's talk about the hepatic VLDL synthesis and secretion. How VLDL synthesis secretion is regulated. First of all, sources of fatty acids that are used for synthesis of triglyceride in VLDL in the liver. We mentioned this before, right? De novo lipogenesis from carbohydrate. For example, under the fat condition, right, we ingest a lot of carbohydrate from the diet, and glucose being absorbed into the blood, right, and then glucose will be transported to the liver, and liver converts glucose to fatty acids, and these fatty acids be reacidified to triglyceride and packed into the VLDL. So this is one source of the triglyceride or or fatty acid triglyceride that in the VLDL comes from diet, right? Basically, it's converted from the carbohydrate, the de novo lipogenesis. And the second sources of fatty acids in the VLDL is the uptake of free fatty acids from the circulation. For example, if we consume a high-fat diet, so we have a lot of fatty acids, right, in the circulation that initially from the diet. And this fatty acid can be directly picked up by the fat tissue, by the liver, and converted to the triglyceride. So this is the second source. And also, during the starvation, the fatty acids can be also picked up from the circulation. And this fatty acid initially is from the fat tissue, right? Fat, mobilization, triglyceride is increased during the fasting condition. So fatty acids released from fat tissue, and that's also causing the increase of fatty acid level in the blood. So liver also picks fatty acids and converted to triglyceride and packed into the VLDL. And this VLDL basically eventually transport the triglyceride from the liver to muscle and other tissues and or utilized as energy, right? And the other condition is like disease condition, like diabetic patients, right? I mentioned before, diabetic patients usually have high levels of fatty acids because of increased lipolysis, right? Due to insulin resistance. So diabetic patients also have a high levels of fatty acids, and this fatty acid can also be picked up by the liver and converted to the triglyceride. So compared to chylomicron, the VLDL triglyceride seems more complicated, right? I mean, you have multiple sources. And chylomicron is only from the diet, triglyceride, right? So enhancement of a synthesis secretion VLDL. So under the following conditions, the VLDL synthesis of secretion can be increased. The first, the fat condition, right? Particularly when we consume a high carbohydrate diet. And second, the high levels of circulating fatty acids. If we have a high level of circulating fatty acids under the which conditions? Do you guys know? When our body will have a higher levels of fatty acids under fasting, right? And what about the disease conditions? Diabetic, right? Diabetic conditions. So, and also we consume high-fat diet, right? To consume high-fat diet during the fasting or diabetic condition, and under these three conditions, that the circulating fatty acids level, are higher, right? Yes, question.
So, this is enhanced during both the fast and fed state, from what you just said?
You mean the high circulating fatty acids?
Yeah, because you said that's during fasting.
Right. So the first condition was like fat condition, right? When we consume high carbohydrate diet, and we have increased lipogenesis. And the second actually high levels of circulating fatty acids. And under this condition, we have a sort of three, situations, right? Does that make sense? The fasting condition, high fat diet, and diabetic conditions, right? Kind of confusing? Yes. Okay. Well, so you guys clear about the first, right? fat condition, right? And the second is high levels of circulating fatty acids.
And that's in the fasting, you said?
Right.
Because your triglycerides are breaking down fatty acids?
Yes, yes, yes. So under the second conditions, we have 3 situations, right, that can cause increase the circulating fatty acid levels. First situation, fasting, and second situation, the high fat diet, consuming high fat diet, and last is the diabetic condition, diabetic patient, right? And then, the digestion of the ethanol. So that also can cause the VLDL secretion. Okay, and then the high concentration of insulin or low concentration of glucagon, basically similar like, under the fat condition, right? So increase the fatty acid synthesis acidification and decrease fat oxidation. So under all these conditions that the synthesis and the secretion of VLDL is increased, right? Is that clear? Right. Okay, maybe I can revise this slide, put all the situation that I just described, then make it more clear, right? Okay, now fatty liver disease. So fatty liver is basically is because of have too much triglyceride lipids accumulate in the liver, right? And this could be caused by the imbalance between triglyceride formation or export, right? So triglyceride in the liver, there are actually 2 main pathways, right? Synthesized or secreted export. So synthesized basically like, lipogenesis or triglyceride synthesis, and export is basically depend on the lipoproteins, right? A lipoprotein is needed to export triglyceride, right? So if this 2 main pathways is unbalanced or imbalanced, then we're causing fatty liver disease. For example, either you have too much triglyceride synthesized, or you have less triglyceride is exported, right? So and eventually we're causing the triglyceride accumulation in the liver, called fatty liver disease. So what are the possible defects that can cause a fatty liver disease? As I just described, the balance, right? The synthesis or export. Sorry? Alcoholism? Yes, I mean, the fatty liver actually divided into 2 different types. One is called non-alcoholic fatty liver, and the other is alcoholic fatty liver, right? So here we're basically talking about the non-alcoholic fatty liver. Yeah, you're right. I mean, so either increase the triglyceride synthesis in the liver, or decrease the export of triglyceride from the liver. So yes, I just said fatty liver the 2 different types, right? Non-alcoholic or alcoholic. So for the non-alcoholic fatty liver disease, then 2 main types based on the cause, right? So cause for the first type, for example, increased triglyceride synthesis. There are some conditions that can causes increased triglyceride synthesis. First is increased levels of plasma-free fatty acids, right? We just talked about it. For example, diabetic conditions, we have a higher levels of circulating fatty acids, and this fatty acid can be taken into the liver and for synthesis of triglycerides. So you have more triglyceride synthesized from circulating fatty acids, right, and causing the increase the triglyceride synthesis. And also the chronic intake of a high-fat diet. If we always, eating high-fat diet for a long time, then it also can cause fatty liver disease. Yes.
Does that depend on the type of fatty acid, saturated or polyunsaturated, or is it any type of fat?
Oh, that's a good question. Yes. And basically it's a saturated fatty acid, right? So saturated, high-fat diet basically is high saturated fat diet, right? Okay, so the second type, the cause for the second type of fatty liver disease is decreased triglyceride export. So some conditions can cause the decreased triglyceride export. Triglyceride synthesized but cannot be, removed from the liver. And for example, blocks in the apoprotein synthesis. Remember, the apoprotein is very important, right? For synthesis of VLDL, for example. If VLDL cannot be efficiently synthesized, then the triglyceride cannot be, removed from the liver, right? And then secondly, the block in the synthesis of lipoprotein from lipids and apoproteins, and basically similar to the first reason, right? The VLDL cannot be synthesized, and lacks of a phospholipid provision. Remember, the phospholipids is important, right, for the lipoproteins, for the lipoprotein, the surface, the monolyl phospholipids. So phospholipids, if some reason that you cannot synthesize enough phospholipids, so the lipoprotein cannot be formed, right? And then the impaired of secretion of lipoprotein. So, and you can synthesize lipoprotein, but for some reason, maybe some, apoprotein lacks apoprotein or some other reasons, that lipoprotein cannot be secreted from the liver into the circulation. So all these factors that can cause the decreased export of the triglyceride, right? Now, alcoholic fatty liver disease. So alcoholic, like if you consume the chronically consumption of alcohol, can impair fatty acids oxidation and increase lipogenesis. So basically, is due to increased triglyceride synthesis, right? The alcoholic, the fatty liver disease. So alcohol, ethanol can be metabolized by this enzyme called alcohol dehydrogenase, and this reactions produces NADH. So NADH, the alcohol metabolite can produce a lot of NADH, right? So this NADH molecules can compete with the NADH that produced during the normal metabolic process. So particularly like fatty acid oxidation process. So because of this competition, so can inhibit or decrease fat acid oxidation, and so fatty acid cannot be oxidized, and then the other alternative pathway will be synthesized right? The triglyceride. So this sort of the simple, the mechanism for how alcohol can cause the fatty liver disease. Basically, decrease fatty oxidation, increase fatty triglyceride synthesis. Okay, so now let's talk about cholesterol homeostasis, right? This is more related to the atherosclerosis. And cholesterol basically present in both plasma circulation as well as and the inside of cells, right? So cholesterol are present in the blood as a LDL, right, in the form of LDL. So increased plasma LDL cholesterol is mainly associated with decreased uptake by the cell. So in other words, if we have a higher levels of LDL cholesterol in the blood, then most likely is because of our body has some problems that cannot be efficiently take up the LDL from the circulation into the cell, right? So something wrong in the receptors. So remember LDL receptor, that is very important, controlled uptake of LDL from circulation into the cells. So couple of reasons that causes the LDL receptor deficient or the defect. First is a decreased LDL receptor number. And for some reason, maybe LDL receptor gene expression is decreased, so you cannot synthesize enough LDL receptor, on the cell surface. Secondly, it could be also due to the defective ApoE or ApoB100. Remember, the ApoE, ApoB100 are required, right, for the binding of LDL to the LDL receptors. So if some reason that we cannot synthesize enough ApoE or ApoB100, then then LDL cannot be recognized by LDL receptors. Even you have a normal amount of LDL receptor, right? But because of it, LDL cannot be recognized by LDL receptor, that also causing the more LDL that remain in the circulation. So again, the LDL receptor is a primary factor that regulates the circulating LDL, plasma LDL levels, right? Does that make sense? Okay, and reason can be decreased LDL receptor numbers, and also maybe some defects in the ApoE or ApoB100 protein, right? Any questions? Yes.
Is there a connection between diabetes and decreased LDL receptor?
Well, not diabetes, but like atherosclerosis, cardiovascular disease.
Great. I feel like I've learned it somewhere, but I'm trying to connect it.
Yeah,
Remember
yeah.
what it was.
Yeah.
So, are you going to talk about?
Yes,
Oh,
yes,
okay.
atherosclerosis. Yeah, we're going to discuss this later. Yeah.
And then is that related to the increase in triglycerides or
You mean LDL receptors?
yeah?
Not much. I mean, doesn't seem that doesn't actually affect a lot of the triglyceride level, but the cholesterol, LDL cholesterol level.
Right, but if you have like an increase LDL,
Uh-huh.
do you have an increase like triglyceride
No, no. So ApoC2 basically, affected the triglyceride levels, right? Yeah. Yeah. So I hope you guys remember that 3 apoproteins very important: ApoC2, ApoE, ApoB100, and if they are defects, it can have different consequences, right? ApoC2 deficiency basically affect the triglyceride level, and ApoE or ApoB100 deficiency normally affect the LDL cholesterol levels, right? And the cholesterol level change is closely related to the atherosclerosis, right? The development. Let's see. I'm thinking if I need to finish this before we do writing. Informal writing. Let me see. Okay, so let me just try to finish this slide. So last slide we talked about the plasma cholesterol levels, right, regulation. And now we also know that cholesterol is also present in the cells, which is directly related to the atherosclerosis development. So inside of cells, there are actually a so-called a free cholesterol pool. And this free cholesterol pool is also keep kind of in balance with the cholesterol esters, right? So several things that can cause the increased free cholesterol inside of cells, which is not good, right? Because of problems. So I listed here actually 5 different things that can make free cholesterol levels inside of cells high. For example, an uptake, right? So this is represent the cells, right? The cell and the cell membrane has the different type of LDL receptors. So if uptake of cholesterol from the LDL, so LDL uptake is increased, then you will cause the increase of the free cholesterol inside of the cell, right? And the second, synthesis. And cells can also synthesize cholesterol. If cells synthesize more cholesterol, that also contributed to increase the free cholesterol pool inside of the cell, right? Does that make sense? So this cholesterol synthesis increase and also increase the free cholesterol pool. And third, the hydrolysis of cholesterol esters. So remember this ACAT is the enzyme that hydrolyzes cholesterol, right? I'm sorry, it's a cholesterol acidification. And the other enzyme called cholesterol ester hydrolysis and catalyze the hydrolysis of cholesterol. So convert the cholesterol ester to free cholesterol. So if this pathway is also increased, or maybe this pathway is decreased, if ACAT is decreased, then free cholesterol cannot be acidified to cholesterol ester, right? Or if this hydrolysis increased, like cholesterol esters, more cholesterol ester be hydrolyzed to free cholesterol. So this also can contribute to the cholesterol pool, free cholesterol pool. And efflux of free cholesterol. So free cholesterol is inside of the free cholesterol can be picked up, remember, by the beta HDL, right? To remove the free cholesterol from peripheral tissue to the liver. And this is a transport protein, ABCA1. It's very important that can transfer free cholesterol from the cell and upload it to beta HDL and then, and eventually to the liver for disposal. Remember this, right? So this pathway is also important that contribute to the free cholesterol level inside of cells. If efflux of free cholesterol, this pathway is decreased, then you will also have an increase, cause an increase free cholesterol inside of cell, right? And lastly, the utilization of free cholesterol. So free cholesterol can also be utilized, for example, for synthesis of a steroid hormone. And this utilization pathway is somehow decreased, and you also causing the accumulation of free fatty acid, free cholesterol inside of cell. Right? So try to remember these 5 different things that regulates the cellular cholesterol levels, right? Which is also important, for the development of atherosclerosis. If this happens in the macrophages, right? And I think we already talked about this, right? The LDL receptor, can control the uptake of LDL. All right, so I just want to stop here.